Human cancer viruses. Human cancer viruses can be divided into two main groups, DNA viruses and RNA viruses. DNA viruses includes human papillomavirus and Merkel cell polyomavirus from the group of non-enveloped DNA. Epstein-Barr virus and the human herpes virus 8 from the group of herpesviruses, enveloped DNA viruses. And hepatitis B virus is also group of enveloped DNA viruses. These are the main human cancer DNA viruses. RNA viruses includes mainly hepatitis C virus and human retroviruses, especially human T-cell lymphoma virus, HTLV. Now we will start to have a brief definition of the oncogenic effect of these viruses. First, we start with DNA viruses, human papillomaviruses. Human papilloma. Sometimes we see integrated copies of human papillomavirus DNA present in cervical cancer tissue, though HPV DNA generally not integrated. It is called episomal in noncancerous tissue, while in cancer we see it integrated into the human DNA to exert its effect. These viral early proteins E6 and E7 are the oncogenic protein. They are synthesized heavily in cancer tissue. These are HPV transforming proteins. They are able to complex with tumor suppressor genes, which are retinoblastoma, RB, and P53. These genes are normally present in human body and cells in the DNA, but when they suppress any transformation of the cell, so-called tumor suppressor genes, when they are suppressed by these E6 and E7 oncoproteins of HPV, this leads to oncogenesis and transformation of the cervical cells, lead to cervical oncogenesis and cancer. These are stages. This is normal cervix to the left, and in the middle, late stage cervical cancer. We see the cervix started with cancer. Then, lastly, to the right, cervical cancer thus spread to the surrounding of the cervix. This is seen also human papillomavirus in cervical cancer. This is the cervix to see where is the position, and below it is the vagina, and above it is the uterus. So, patient examined by speculum examination see to the right how is the cancer started. Cervical cancer by human papilloma develops very slowly, sometimes taking years to decades, عشرات السنوات. Multiple factors are involved in progression to malignancy. However, the most important is persistent infection with high-risk HPV types, which is very necessary components for the process of oncogenesis, mainly types 16 and 18. And there is a vaccine nowadays given for the women to prevent this, and this is divalent vaccine, contains L1 protein, late protein, for human papillomavirus for mainly types 16 and 18 to prevent cancer. Epstein-Barr virus. EBV linked to Burkitt lymphoma and nasopharyngeal carcinoma, and also to Hodgkin's disease. These tumors usually contain EBV viral DNA, both integrated and episomal form. Could be integrated and episomal. By the way, EBV is the first human oncogenic virus discovered in the 50s, I think so. EBV encodes viral oncogene protein اللي هو LMP. LMP1 latent membrane protein mimics activated growth factor receptor. When this receptor acts as activated, they give signal to the cell to continuing growth. Also, several EBV including nuclear antigen, EBNAs, called EBNAs, EBV nuclear antigens, are necessary for immortalization of B cells because EBV enters or infects B cells only. So EBV main oncogenes are LMP and EBNAs. Let us see these tumors, Burkitt lymphoma and nasopharyngeal. This is Burkitt lymphoma, mainly in African boys, commonly, and this is nasopharyngeal carcinoma. Nasopharyngeal carcinoma caused by EBV, but not always. This is the location, up is the location of the nasopharyngeal carcinoma, and below is the cancer by examination through the nose. Human herpesvirus 8, they were called also Kaposi sarcoma virus discovered with HIV because high association between this virus and HIV, and it mainly infects male homosexual males that have HIV. Kaposi sarcoma-associated herpes, also known as human herpes 8. It is not ubiquitous as most other human herpes viruses. Ubiquitous يعني commonly distributed and highly seroprevalent. It is not. It is a rare virus. It is believed to be the cause of Kaposi sarcoma. Nowadays also primary effusion lymphoma and lymphoproliferative disorders, Hodgkin's disease. Kaposi sarcoma herpesvirus has a number of genes related to cellular regulatory genes that stimulate cell proliferation and modify host defense mechanism. So it affects host defense mechanism and cell proliferation. This is picture of man has Kaposi sarcoma due to human herpes virus 8. Now the fourth DNA virus, hepatitis B virus. Epidemiological and lab studies have proved persistent infection with hepatitis B to be an important cause of first chronic liver disease, of course, and second hepatocellular carcinoma, HCC. HPV considered a carcinogenic virus. The mechanism of oncogenesis remains obscure. Persistent viral infection leads to necrosis, inflammation, regeneration. This leads to cirrhosis, then hepatocellular carcinoma. 8 HPV genotypes are present, designated A to H. HPV genotypes C and B, mainly associated with high risk of hepatocellular carcinoma, genotype C and B. The risk of developing HCC directly proportional to the hepatitis B DNA level. يعني بنسوي quantitative real-time PCR for hepatitis B. We see the higher the DNA level of hepatitis B, the higher the risk of hepatocellular carcinoma. There will be, and this lead to hepatitis B DNA integration into the host genome, leading to insertional mutagenesis. So there will be mutation due to insertion of the DNA integrated. It's called insertional mutagenesis in cancer-related genes. So causes a mutation in cancer-related genes, especially tumor suppressor genes, lead to inhibition of them, mutation in them. Integration of HBx. HBx هو the main oncogene hepatitis B virus. Main oncogene of hepatitis B called HBx It was called HBX because this gene, first when identified, have no role, so they call it HBX X gene. Now, integration of this HBX gene into the hepatocytes lead to inhibition of p53 tumor suppressor, decrease the expression of p53 activators also, and inhibition of retinoblastoma B RB tumor suppressor gene. So inhibit both p53 and RB genes and inhibit p53 activator. These three main mechanisms of action of HBX gene when it's integrated into the hepatocytes, into the genes of hepatocytes. This picture shows multiple mechanisms could cause hepatocellular carcinoma, but the most common hepatitis B and hepatitis C. We see a normal liver, then becomes cirrhosis, cirrhotic liver, تشمع الكبد, then ends with end-stage cirrhosis with hepatocellular carcinoma. Cirrhosis is the pre-malignant condition, can be regarded as pre-malignant. This is a picture from hepatitis C virus. We will talk about this now. Now we end with DNA cancer viruses. We start with RNA viruses, oncogenic RNA viruses. The first one, hepatitis C virus. Hepatitis C virus genotype 3 infection had increased risk of HCC. Hepatitis C virus may have direct effect or indirect effect in carcinogenesis. Start with indirect, which is more common. Patients with advanced fibrosis or cirrhosis are at increased risk for carcinogenesis because of the chromosomal alteration that occur in the fibrotic tissue. Even there will be also chromosomal alteration in fibrotic tissue lead to tumor formation. Direct effect. They found that one of the non-structural genes of hepatitis C virus promotes fibrosis, either through first increase TGF, transforming growth factor beta, and B, activating hepatic satellite cell formation. There are hepatic satellite cell, they will be start proliferating and causes fibrosis and then cancer, or inducing transforming growth factor beta. These are all induced by fibrosis that is induced by hepatitis C virus non-structural proteins. Now we go to the human T lymphotropic viruses, HTLV. HTLV viruses, another RNA viruses, but it is a group of retrovirus. This virus, HTLV, is related to HIV. Also, it is a retrovirus. This picture of HTLV, and this is an EM picture of HTLV. It is also enveloped, just like HIV, and it is RNA virus, positive strand, but it have reverse transcriptase. HTLV group of retroviruses, 1 and 2 there. One has been established as a causative agent in adult T-cell leukemia and lymphoma. Then HTLV1 established as causative agent, well established as causative agent of adult T-cell leukemia lymphoma, ATL. HTLV1 is expressed at very low level in infected individuals. Unlike other viruses, when they are oncogenic, they will be high level of these viruses, like as we said, hepatitis B and also hepatitis C. But HTLV1 is expressed at a very low level. By qPCR, we see that the DNA is very low in infected individuals. HTLV have marked affinity for mature T cells. EBV loves B cells. HTLV from a cell, its name mature T cells. It have a gene called trans-activating regulatory gene. TAX, believed to be necessary for viral replication and this may contribute to oncogenesis by modulating cellular genes that regulate cell growth. So effects on oncogenesis are a trans-activating regulatory gene. Trans-activating regulatory gene have oncogenesis by modulating cellular genes that regulate cell growth. Transmission of HTLV. Two main modes, and it has unique properties in transmission. Transmitted from mother to child by breastfeeding. By breastfeeding, it is very important mode. In early life, infection by this virus increase risk of adult T-cell leukemia and lymphoma. Infection in early life by this virus in children by breastfeeding is increase the risk of ATL, lymphoma and leukemia. Second mode of transmission by blood transfusion. It is effective means of transmission of the virus, blood transfusion. And thank